So for this audio lecture, we'll be focusing on Chapter 18, and this is the second audio lecture for Chapter 18, and this one will focus on cell death. Okay, and as we see in this figure below, we can see how the arrows are indicating cells that are undergoing cell death, more specifically apoptosis. So we do want to be aware that there are two different terms related to cell death, apoptosis and necrosis, and so we'll talk about those next. And so we want to keep in mind that apoptosis and necrosis are not the same, to answer that question. So necrosis is going to be general cell death, or sometimes termed uncontrolled cell death, where apoptosis is a programmed cell suicide. And so just in those brief kind of definitions of them, we can get this idea that apoptosis is a controlled process that cells will undergo, okay? Where necrosis is a more uncontrolled effect that happens. And so we can look at the processes that are happening between these two different pathways. Apoptosis is not going to be damaging neighboring cells because the cell is actually going to shrink. We're not going to get inflammation occurring, and we get these apoptotic bodies that are formed. So the cell's not being the cell's not lysing like we see in necrosis. And so with necrosis, when you get the cell lysing, you get lots of different factors and material being released that can cause damage to neighboring cells. Okay, and again, it's a very programmed, kind of cell signaling pathway that happens. So this needs to be activated in order for that to be activated, in order for apoptosis to happen. And so again, we could compare the two processes. The focus of this lecture is going to be on apoptosis when we're looking at cell death. So what are some of the hallmarks of apoptosis? If we look at the cell, for the morphology, they become really rounded and small and refractorial. And so if we think back to that first image is where we have the arrows indicating, you can see that those cells have a very round and small appearance to it. So there's actually this like shrinking that's going to happen. Another hallmark of apoptosis is fragmented DNA. And so you can see on the gel on the right of this slide, how we can see how over time we see that fragmenting of the DNA, almost like a DNA ladder that's formed by these thymocytes, the lymphocytes from the thymus of a mouse. And this mouse these cells have been induced to go under apoptosis through the anti-Fas death receptor. Okay, so what can be done is known as a tunnel assay. So a tunnel assay looks at these fragmented DNA. We also have different pro-apoptotic factors that can become activated. Another hallmark of apoptosis is a loss of mitochondrial membrane potential. So keep in mind, in order to produce ATP, we need to have that membrane potential to allow for hydrogen ions out to then flux back in. And so if we use positively charged dyes, they don't get incorporated in the mitochondria like they would do. There's also a loss of cytochrome C from the intermembrane space. And again, this could be done through detection through antibody staining. And phosphatidylserine actually will end up flipping. So if you remember back when we were talking about the cell membrane, the phosphatidylserine is unevenly distributed on the cell membrane, and so this can get detected by using fluorescent labeled annexin 5 to look at the incorporation of that. So cytochrome C gets lost because it gets released from the mitochondria, and this initiates and becomes a key driver for apoptosis. Okay. So if you look at this figure here, in the upper panel, in panel A, we can see our control. We can see how the cytochrome C that's labeled with GFP, the green, co-localizes, it overlaps in the staining pattern of the mitochondrial dye. It'd be nice if they had another column where they had the yellow, right? In the lower panel B, which is UV treated to induce apoptosis, you can see how we have the cells there that have a diffuse staining pattern for cytochrome C, that it's no longer localized just with the mitochondria. Okay, so again, the change in the staining pattern lets us know that the cytochrome C has changed its distribution pattern and has initiated this intrinsic apoptosis cascade. So up on our D2L files, there's a movie on apoptosis, so I'd recommend viewing that to see the different steps that you can see through the microscopy. So there's a number of reasons for apoptosis to occur. So there's both physiological and developmental roles for apoptosis. So we'll look at a couple different examples of apoptosis. So for neutrophils, they can undergo apoptosis because we never know when we're going to face an infection, right? And neutrophils are one of the immune cells that are used to fight an infection, especially bacteria. And so we're continuously having to produce neutrophils, but if there's no infection present, we don't need those neutrophils, and they don't stick around very long. So they're kind of like, if you left a dairy product on the counter on a hot day, you wouldn't want to eat it. So you don't want old neutrophils trying to fight an infection. And so by programming them to die, it actually allows the bone marrow to then produce new fresh neutrophils. So if there's no infection present, those unused neutrophils will actually be programmed to die. When we look at metamorphosis of different organisms, especially like tadpoles, we can see how cells will undergo apoptosis to kind of remodel the tissue, right? So as a frog develops from a tadpole into a frog, they lose the tail, and that tail actually goes apoptosis. Okay, the figure that we see here actually is showing digit sculpturing that's undergoing in mammals. So you did this. Mammals do this. So for our hands, they're actually interconnected during embryonic development, but then the cells that are in between our fingers actually undergo apoptosis. So we can see here in panel A where the cells are taking up our dye. So we can see those cells present, but as the cells initiate apoptosis, they no longer take up the dye, and we can see the area where we don't have the staining in between the fingers. Okay, and this is just after the time difference between panel A and panel B is 24 hours, but again, we could take a picture of you in fetal development and you would have these webbed fingers, but yet the majority of us are not born with webbed fingers. So obviously if there's a defect in apoptosis, this webbing may still be present in individuals at birth. So apoptotic cells end up getting engulfed either by their neighbors or by macrophages. So macrophages, again, another immune cell, and they're important for removing material. So again, an apoptotic cell will actually display eat me signals, where normal cells will actually display a do not eat me signal on their cell surface. So again, if a cell undergoes apoptosis, they change those signals, and some of that is that flipping of phosphatidylserine, and that actually says to macrophages to eat me. Okay, so that the macrophage would actually come in and engulf. So here in our panel, we can see in panel A, we have an apoptotic cell that's in vitro, but in panel B, we have this apoptotic cell that's in vivo, and it's getting engulfed by the phagocyte, so it's actually being internalized. Now, to compare that to a necrotic cell, in panel C, we can see our necrotic cell where it's actually just ruptured. So again, as I mentioned before, necrotic cell, all that material is being released into that area. Neighboring cells then are potentially being affected by that. So apoptosis can be initiated by two different pathways. There's the extrinsic pathway, which is due to Fas ligand that is produced by lymphocytes. So a lymphocyte, when it recognizes that a cell is in danger, will produce Fas ligand that will bind to the receptors on the target cell. It will initiate receptor tyrosine kinase signaling that then will lead to cell death, to apoptosis. There's also intrinsic cues, and so this is a balance between anti-apoptotic factors and pro-apoptotic factors that, again, the cell, just like in any cell signaling, will take in all that information and then decide. And so if there are higher amount of pro-apoptotic factors, that would lead to the cell going into apoptosis, but this can also be blocked by anti-apoptotic factors. So both the extrinsic and intrinsic apoptotic pathways use intracellular proteolytic cascades. And what that means is that they're going to use intracellular signaling in order to then initiate the cleavage and breaking down of material. So proteolytic breaking down, okay? So for the intrinsic pathway, we're going to get the release of cytochrome C from the mitochondria. Remember, we saw that figure where when we stained for cytochrome C, or we looked at the GFP, we ended up seeing it went diffuse. It wasn't just located, co-localized with mitochondria. This causes the assembly of an apoptosome and a caspase activation. Caspase are found in a proform. They then become active, and this causes a cascade of caspases becoming activated. Caspase 9 is one that's commonly looked at for activation. And again, this will signal that we have apoptosis occurring. So for our extrinsic pathways like Fas, you're going to end up getting the Fas death receptor, and this directly leads to caspase activation. And there is also the EphA7 receptor, which is a tyrosine kinase receptor, and that leads to caspase activation. So again, we can look at caspase activation as an indicator whether we have apoptosis occurring, and we can also look at the location of cytochrome C to indicate whether we have intrinsic, and then we can look at usually Fas ligand, Fas receptor activation as extrinsic. So what ends up happening is that we have our procaspase, which we see in panel A. That is end up going to be cleaved. So the prodomains come off, and so now we have an activated caspase. So this is one of the ways that we can regulate whether apoptosis is going to occur, because we have a proform and we need to cleave it in order for it to become activated, right? So it's not that we have our cell with activated caspases already in there. That then initiates a cascade. So we can see in panel A where we have this one molecule of active initiator caspase, and then that further gets amplified down as additional caspases end up getting activated. And then we get cleavage of different factors within inside the cell, like the laminar, the nuclear lamina, and cytosolic proteins. So initiator caspase generally are caspase 2, 8, 9, 10. Execution caspase are 3, 6, and 7. So generally researchers will look at caspase 8 or 9. And then caspase 3 is another one that's commonly looked at, either through Western blots or immunofluorescence, and being able to detect the pre versus cleaved form. So I just do want to note that the newer edition of our textbook has taken panel A and panel B and merge them together so that it has a more coherent displaying of the information. And so though the 4th edition of the textbook has these as different pieces, keep in mind that one activated caspase molecule is going to end up cascading and amplifying the signal. Okay. So when looking at intrinsic apoptosis, we had mentioned that cytochrome C is going to be released from the mitochondria. So we see here in this figure how apoptosis signal happens. Cytochrome C is going to move from the intermembrane space of the mitochondria and be released. It's through activated back molecules that this happens. That cytochrome C then binds to adapter proteins, and these assemble together into a complex. Okay, so APAF or APAF1 is an adapter protein that will bind the cytochrome C in complex and assemble together. We then have a procaspase 9 that becomes activated, and this all assembles together into this large complex that is known as an apoposome. So this further cascades with caspase activation and then leads to apoptosis. So contrasting this with the extrinsic apoptosis, what we'll end up getting is these fast death receptors that are going to aggregate together. They're going to bind to the fast ligands that will be on the lymphocytes, as we can see here. This complex is with the FAD adapter protein, which has a death domain that's going to bind to the receptors, and a death effector domain that will end up binding to our caspase. This all complexes together to form what's known as the DISC, death-inducing signal complex. This will end up leading to the caspase being activated, so we can see how we get activation of the caspase, and this would further cause activation of additional caspases so that we end up with an apoptotic target cell. So again, comparing intrinsic is the cell telling itself we need to die and providing those signals to the cytochrome C, where extrinsic, another cell, the lymphocyte, is telling the cell you should die. So you're going to go into apoptosis. So the stimuli that actually releases the cytochrome C in the intrinsic apoptosis pathway can be activated through different responses. So we had mentioned in our previous audio lecture on cell cycle about P55 activation. So if we have unrepairable DNA damage, whether that's because of the telomere lengths or whether exposure to UV or some kind of DNA damaging chemical, then this can't be repaired, then it would be best for the cell to go into apoptosis, right? And so with P53 activation, this is one way in which the intrinsic apoptosis pathway can be activated, and then leading to cytochrome C activation. So the BCL proteins help to regulate the intrinsic apoptosis pathway. So there are anti-apoptotic BCL2 proteins, which we can see in panel A. There are pro-apoptotic proteins, the BH123 proteins, back and backs. And there are pro-apoptotic BH3 only proteins, such as BAD, BIM, BID, PAM, and NOX. Okay, so again, this allows us to have regulation both in an anti-apoptotic way and a pro-apoptotic way. And so these different genes, these different proteins allow for this regulation. So BH123, it will actually aggregate together upon apoptotic stimuli. That aggregation allows for the cytochrome C release from the mitochondria, as you can see in this figure. Activation of BCL2 will block the aggregation, so it prevents the intrinsic pathway from being activated. So it's going to prevent the cytochrome C from being released from the mitochondria, as you can see in this figure. So BH3 only proteins will inactivate BCL2, so that actually will promote apoptosis. So BH3 only proteins, as you can see from our figure here, it's going to inhibit, it's going to inactivate that anti-apoptotic BCL2 protein. So it's not going to be able to block that aggregation. So your BH123 proteins are still able to aggregate together, allowing for cytochrome C release. So some viruses will produce proteins that inhibit the intrinsic apoptosis cascade. So keep in mind, if you're a virus, you don't want the cell that you're infecting to go into apoptosis, because then you can't replicate. So they'll inhibit apoptosis or produce IAP proteins. And so those IAP proteins will block activated caspases from being able to bind and cause that cascading, or they could block your BH123 proteins from aggregating together, allowing for cytochrome C release. So again, it's a mechanism that allows the virus to be able to not survive because they're not really living, but allow the host cell to survive so that the virus can use its machinery to make copies of itself. Now, our cells can also produce anti-IAP molecules. So anti-IAP molecules are going to inhibit those inhibitors of apoptosis, so that apoptosis would end up happening. Okay, so here we can see how these anti-IAPs are going to block the IAPs so that we still get activation of our caspases and our execution caspases, so we end up getting apoptosis. So this is more characterized in Drosophila. It's not as well characterized in mammalian apoptosis. So extracellular survival factors can also inhibit apoptosis. So if we have a survival factor, it's going to bind and activate its receptor. We'll have downstream activation and cause production of these anti-apoptosis proteins or inhibitors of apoptosis, so that the end result is that we end up getting apoptosis blocked. Okay. So there's chromosomal translocations that can cause excessive BCL2 production, and this is found in many B cell lymphocytes, so it blocks the apoptosis from occurring. So an example of how these extracellular survival factors can inhibit apoptosis is shown in this figure. So here we have these nerve cells. The axons are attached to the target cells. Target cells are going to produce survival factors, and so this would inhibit the nerve cells from undergoing apoptosis. But if there's too many nerve cells, there's not going to be sufficient survival factors being produced to support all of them, and what will end up happening is that some of these nerve cells would end up going into apoptosis. So the idea is that it's better to overproduce the number of nerve cells, so that then we can decrease the number of nerve cells. And it ensures that by having an overproduction, that every target cell is actually going to have a connection to a nerve cell. So if we only produced, 3 nerve cells to begin with, the likelihood that the connection by that axon would be to all 3 target cells is not very favorable. So if we actually overproduce the number of nerve cells like we see, where we have like 6 of them, we guarantee that we have a connection to at least one connection to each target cell. So when the target cell is producing survival factor, then it would allow the ones that actually are able to take up the survival factor to survive. The other ones would be initiated to go into apoptosis. Okay, so again, this is some of that remodeling that's going to happen due to apoptosis. And so again, we see this, if you take a developmental biology class, a lot of that, the changing of the tissues through development, apoptosis is going to guide that. Okay, so keeping in mind that apoptosis is controlled, we can see how there's different steps of regulation, right? We can have regulation in the fact of a signaling molecule, so survival factor blocking it, but then also needing ligands being triggered, or we need to have these pro-apoptotic factors to be present in order to get the cytochrome C release. Well, after cytochrome C release, we need adapter proteins to come together, then we need activation of caspase and a cascading of caspase in order for that to happen. So every one of those steps is a level of regulation to ensure that only the cells that we want to die end up going into apoptosis. And again, whether this is because of remodeling, this is because the cell has become infected by a virus, it allows us as the organism to control which cells undergo apoptosis and are removed in a controlled fashion. Okay, so this concludes the audio lecture for apoptosis, but in more general audio lectures for chapter 18. So again, as always, if you have any questions, please do not hesitate to contact me, and there'll be some assignments that will be posted on our D2L class in order for you to complete to assess your level of understanding on this information. So thank you.